Let's continue with this lecture. So this part 2 of our chapter 11, and we're looking at cells in the nervous system. Keep in mind that usually when we talk about these cells, we have a functional component of the cells, which are the neurons. And then more often we have support cells called neuroglial cells that are usually a lot more common, greater in numbers, and has a separate set of functions for each of the neuroglial cells that we'll discuss coming up. So in terms of neurons right here, it's probably easier to review those when we look at a picture. So in terms of parts of the neuron, we'll have the biggest area of the neuron. It's a cell body or the soma. So those words are used interchangeably. Soma is the cell body, and this is where we'll see the nucleus. This is where we see all the major organelles. Now we will see some organelles as well in the axons, right, and in the dendrites, but most of the main functional component is going to occur, right, and main repair and healing component is going to be due to the drive of the nucleus, and that's found in the soma or cell body. Now, the nucleus being nice and round, the soma should be very circular as well, and keep in mind that when we have a cluster of these cell bodies in the central nervous system, that cluster of cell bodies we call a nucleus or nuclei. And when the same cluster of cell bodies are found in the peripheral nervous system, it can be a millimeter away from the spinal cord. Doesn't matter. It's not inside the spinal cord. We call the clusters of cell bodies a ganglia. Now, when we're looking at the ganglia or the nucleus, and we're looking at the cell bodies right here, we'll have lots and lots of what we call these dendrites sticking out of the cell body. Usually you have one axon terminal coming out that turns into the axon. The axon itself can split afterwards or downstream of this, but you only have one axon terminal or one axon hillock region where one main axon that exits out. You're gonna have lots of spines and dendrites coming out of the cell body, but only one main axon exiting out. Now, if you look at the dendrite right here, there are multiple dendrites coming out, and within the dendrite, you sometimes might see little spikes exiting out. That's a dendritic spines, and it's usually where one axon synapses with another one. Now, when we compare, and usually when we're drawing like a circuit of these axons and neurons, we have like a one neuron making contact with another neuron, which makes contact with another neuron. In reality, this one cell body has lots and lots of dendrites, and lots of these dendrites have lots of dendritic spines. We can have up to 200,000 different axons, different neurons making contact with this one single axon. 200,000 different neurons making contact with this one neuron that's postsynaptic, meaning that it's going to take a lot of action potentials from different neurons to either trigger it or even inhibit it. So just keep that in mind, that it's not a simple one-on-one ratio where one neuron makes contact with another neuron. Usually it's much more complex, where you have thousands of different axons from thousands of different neurons way over here, and it makes contact with one postsynaptic neuron. Now, we have in the nucleus nissl substances, sometimes called nissl bodies, and these are chromatophilic substances. These are where the rough endoplasmic reticulum are. This is the primary site of protein synthesis, and within the site of protein synthesis, you'll notice as well that in order for repair to occur in the axons, you will need functional nissl substances, and obviously you're going to need functional nucleus as well. All right, we talked about the dendrites. They're highly branched, coming off the cell body, and with these dendrites, these are the actual connection to what we call presynaptic neurons. Those presynaptic neurons are the 200,000 neurons that might make contact with this one neuron. The axon comes off, and there's only one main axon that comes off, unlike the dendrites. And with the axons, it comes off at the very start and is connected to the soma by the axon hillock region. The axon hillock region right here then is going to move downstream and turn into an initial segment. these two regions form the trigger zone. So when those 200,000 neurons have somehow meshed and somehow activated and causes depolarization of this postsynaptic neuron. Once we have activated this neuron, then the neuron will trigger an action potential down its axon. And the trigger zone is where the action potential will start and get triggered down the axon as it jumps through all these different nodes around VA to the terminals right here. Again, the trigger zone, site where action potential for this neuron is going to be triggered, and then is going to move downstream from that axon terminal. And then here you see a neuron with Schwann cells right here, and the Schwann cells are going to myelinate, and they're going to ensure that action potentials will just occur on the bare areas. And the bare areas are called the nodes around VA, and we'll go back to that in a few minutes. Now, a couple things about these axons. Number one, axons, we can have movement of axoplasm, which is a cytoplasm, from the cell body towards the terminal. And the movement of these also carry necessary nutrients, proteins for, repair, growth, and renewal. Now, the good thing is this, what we call anterograde movement, allows the axons to stay alive. If you think about these axons and these cell bodies, they're going to be with us throughout the course of our lifetime. So they're always going to be continuously, repaired and heal and then regenerated, especially if the trauma isn't massive. Keep in mind that as long as the cell body is intact, the soma is intact, and as long as the axons, are slightly moved, even if they're offset, if they're split and cut, as long as they're somewhat close in proximity, you can bring them back together and heal that axon. So that is possible as long as the healing of the neuron and healing of the axon is possible, as long as the cell body is intact, as long as the axon is close together. The ends that are split are close together, Now, it can heal because of that anterograde movement of axoplasm that's going to bring with it organelles, proteins, cytofilaments, microtubules, microfilaments. They're also, then you can reincorporate it and bring those two ends of the disjointed axon together. That allows for healing. Now, that anterograde flow is really important. It allows you to connect the cell body to the rest of the axon. There's also retrograde flow. Retrograde flow allows us to get rid of damaged organelles, get rid of damaged fats and all the proteins that's damaged throughout the course of many years of us being alive. We need to recycle all of that. So we need retrograde flow from these axon terminals or anywhere in the axon. And now we're going to bring it back to the cell body where we have lysosomes ready to digest and recycle everything. Again, we don't want any of the trash to accumulate in the axon. There's just not a lot of room. So we need to bring it interiorly and retrograde back to the cell body where we have lysosomes ready to get rid of everything. The bad thing about this is because of the retrograde flow, we can have certain viruses that can enter the central nervous system by this retrograde flow of axons. The best example, rabies. Rabies, when you get rabies, the rabies virus is incredibly deadly. If you have gotten rabies and exhibited signs of rabies, the chance of survival once you've exhibited signs is infinitesimal, It is so small that until the early 2000s with Jenna Giese, The good news is that this happened at the medical college. And Jenna Giese right here, I don't know if it's Gina or Jenna, but with that first instance in the early 2000s, what we saw with Gina Giese was that she was the first person ever to survive, rabies, after showing signs and symptoms. Now, a sign is something you see. A symptom is something somebody tells you. So by the time she entered the hospital, she had been bitten by a bat. Usually in America, bats are the vectors, right? They're how we carry that rabies virus into our bodies. So it's been that bat's been infected with rabies. Now it's been Gina Gysi. She didn't know. Bats have very sharp teeth. So when she's been bitten, she didn't know she was bitten. And then she started hallucinating and she started acting. And, and the big one is usually the hallucinations as well as drooling, right? Once you have this massive drooling, the hallucinations have brought her into the hospital. At that point, she's exhibiting signs. There was basically nobody that's ever survived rabies up until this point, until her. Nobody's ever survived with showing symptoms, right? Now, if you've been bitten by a bat and you can't find the bat, we assume you have rabies and you've been exposed. At that point, you need to go into hospital and they'll give you, right, shots. The shots contain antibodies, immunoglobulins that are there to bind up and remove the virus before it enters your central nervous system. Those shots are incredibly efficient, right? So as long as you get to the hospital in time, those immunoglobulin shots of rabies not the antivenom, but antitoxin, right, is going to bind the rabies virus and prevent the virus from actually going through retrograde flow and entering the central nervous system. Keep in mind, most viruses don't enter the central nervous system, right, and most bacterium as well, they don't enter the central nervous system because we have certain cells that forms a blood-brain barrier. Well, this rabies and herpes virus, they kind of go underneath that, right? They go through this retrograde flow. And for Gina Gysi, she had experienced signs and symptoms. She went in, and the only thing they could do was put her in a medically induced coma and then give her high dose dexamethasone, which is an incredibly strong type of steroid, right? Because at this point, because of the rabies entering the central nervous system, she's been having massive inflammation in the brain. So they need to calm it down. So they give them high dose dexamethasone. Dexamethasone is also the really strong steroid that we give people right now for coronavirus in the hopes that it will decrease the inflammation in the lungs and in the cardiovascular system. So the dexamethasone will hopefully decrease the inflammation in the brain. They put her in medically induced coma. They then give her really strong high dose of antivirals. And if they believe in God, they prayed. And what happened was that she was in a medically induced coma for a couple of weeks. And after they remove her from the medically induced coma, she woke up, right? And she was able to live and survive. Now, keep in mind, when I say she survived, that does not mean that she came out of it exactly the same way. In other words, there were also, right? There was, when she got out, she had to relearn how to talk. She had to relearn how to walk. She had to relearn how to feed herself. So she had to relearn all of the things that for our normal daily living that we take for granted, all because of that initial trauma from the rabies virus. Now, after the rabies virus treatment protocol, they called it the Milwaukee Protocol. And they thought, and a lot of infectious disease experts thought that this was a magic bullet, that we can now start treating people, and for the most part, they should survive on this Milwaukee Protocol. So people with rabies across the world then should then hopefully survive if they're given this protocol after they've developed symptoms. So in America, we don't see rabies much, but in the developing world, India, Thailand, right, Asia, those are all areas where we have endemic rabies. And for the most part, those people in those areas, well, they don't really understand rabies as much, and they wait, and you see lots of deaths because of it. What they found out was that even with that Milwaukee Protocol, for the next 3-4 times they attempted the protocol, all the people that were given that protocol died from rabies. In other words, it somehow only worked for a genome, right? Now, what they've done in the last few years is titrate it, changed it a little bit, right? They still start with that real high dose dexamethasone, antivirals, right? Now they've just kind of changed a mixture of antivirals. And now they've had a couple more people survive rabies after exhibiting signs, but it's still not the magic bullet. So the best thing to do if you've been anywhere near a bat is number one, catch the bat, right? Or best thing is to make sure you're not near the bat, right? Don't get close to it. Call somebody to catch it. Number two, right? Then if you've been bitten, assume that you have been exposed to rabies and go into the ER. Number three, right? In the ER, they should start giving immunoglobulins that will bind to the rabies virus to prevent them from entering the central nervous system. Everybody in the old times talk about how painful it is. It's not painful, right? For the most part, that actual immunoglobulin shot is less painful than the shot that you get for the flu. It's not an intramuscular injection. It is an injection very similar to what we see with insulin, right? It's a subcutaneous injection. You need multiple shots of these, but these are all very, right, small needles subcutaneously administered. Now let's talk about the types of neurons. We can classify neurons based on functions or based on structure. When we classify it based on function, they're either sensory, motor, or interneurons. Sensory or afferent carry action potentials towards the central nervous system, and they carry sensory information from your periphery to the central nervous system. Motor signals are the exact opposite. They come from the brain and the spinal cord and exits out towards your periphery and your limb. They carry motor signals out to your targeted muscle. Now, that motor signal can be going to the heart. They'd be going to your smooth muscles in your stomach. It can be going to glands. In other words, motor signals doesn't have to be going only to the skeletal muscles. It can go to other involuntary muscles. Remember, involuntary muscles and glands also requires some kind of activity to stimulate it. So the motor signals here will stimulate it even though you're not in control consciously. Then the most important one and the most common ones are these interneurons. Interneurons are what we kind of associate with the brain. The thinking, the memory, the processing. That's interneurons' function. Its main purpose is to decipher all the different sensations coming in and then formulate an appropriate response to it. Perfect, right? It's the one that interprets and then figures out an appropriate response for that situation. All right, now we can classify also based on structure. We have multipolar neurons, which we just saw, all right, with lots of dendrites coming off and one main axon. The most common type of neurons in the central nervous system. Most motor neurons are multipolar. You're receiving multiple inputs. Remember, up to 200,000 inputs. And then one main output. That output then will go to another neuron, which will then cause your muscle to move. Perfect. Bipolar, one dendrite, one axon. The dendrites, though, if you take a look, they split almost right away. Right? The bipolar neurons will be mostly seen in regions where we have special senses. So sensory receptors of the eye, photoreceptors, cone cells, right, are a type of bipolar neuron. Olfactory neurons are kind of bipolar neuron, very important in special senses. Next, right, unipolar neurons. In unipolar neurons, like we see here, right, usually we see it in our general sensations. We saw it earlier in the part 1 of chapter 11, right? You have pain sensation, and pain receptors in your fingertips. Those pain receptors are connected, right, to a sensory neuron. Its axon comes all the way down, all the way across. Now, there are no dendrites here, right? So all we have in a unipolar one is a cell body and then an axon that splits in two, right? As it splits in two, one goes to, all right, one part of the axon goes towards our fingertips and carries sensation, right, for pain signals. Then when it goes to your fingertips, it then connects to pain receptors. So when you get a cut, the pain receptors activate, causing an action potential through that axon. There are no dendrites here. It's just central receptor and then an axon connected to it. Now, the cell body here, especially when we talk about, right, your general sensations, the cell body is found as part of our dorsal root ganglia, right? So take a look at how round the cell body is, and now you have a collection of these real round cell bodies outside of the spinal cord. What do we call that? A ganglia. Where do we find it? We find it in the dorsal root region. That's how we get the name dorsal root ganglia. Next, we'll talk about the different type of cells. In terms of cells, know these cells. These are very good for either matching or multiple choice. First cell, astrocytes. Now, astrocytes are these cells right here. They look like little star-shaped cells. These are cells that are neuroglial cells that are there as accessory to the neurons. So you have the neuron right here, the functional component of the brain and the spinal cord, and now you have astrocytes. One of the really important things about astrocytes is that it protects the brain environment. What it's going to do, the astrocytes, is going to have processes that covers up the capillaries found in the brain. As it covers it up, it prevents bacterium. It prevents certain proteins. It prevents material, even some molecules, from getting through by forming the blood-brain barrier. The foot processes then are interconnected to each other from other astrocytes. It covers up any small gaps formed by the endothelial cells, which form the capillary. So a capillary, I just want you to imagine, is just a simple tube, like a garden hose. And instead of the plastic making the tube, it's made out of cells, simple squamish epithelial cells. Why simple squamish? Because they're flat. Flat then allows oxygen and gases to diffuse in and out really easily. Now, the simple squamish epithelial cells that form the capillary has a special name. They're called endothelial cells, right? And within those cells, so you have one cell over here making up this top portion and another cell over here making up the bottom portion. In reality, it's more than one or two cells, right? So what happens is there's tiny little crevices between those cells. Tiny little crevices can allow water to get through, can allow salt to get through, can allow small molecules to get through. But proteins, large proteins can't get through, but medium-sized proteins can. And in the brain, we want to limit what gets in and out of the brain and the spinal cord, right? There's only so much room in your skull. The last thing we want is to have water in your skull or too much fluid in your skull. It will cause you to herniate the brainstem. So to prevent excess movement of any kind of proteins, any kind of even molecules, right? And specifically, viruses and bacteria from getting through, we have these foot processes from the astrocytes. They form tight junctions that prevents even water movement in and out, right? So what happens then is that this blood-brain barrier is very protective. It protects the neurons from toxic substances. At the same time, though, right, it does allow for nutrient exchange. We're going to have glucose receptors in the capillary, right, in the endothelial cells. Then you're going to have, right, a little bit of diffusion from those cells, and it's going to be, a facilitated diffusion of glucose from your plasma out through the endothelial cells, and then they can go into the neurons. But besides glucose, salts, and maybe a little water movement, definitely oxygen can get through, but everything else has a hard time getting through. Now, the best example of how hard it is to get material through is this. in the 1940s and 50s, we kind of understood Parkinson's disease was caused by a lack of dopamine. So the dopamine releasing cells are found in a specific region of the brain called the substantia nigra. So we knew that there was a loss of function in those regions. And usually that at the very start, you just lose dopamine, right? Those cells that are dopamine releasing cells from the substantia nigra are there to inhibit unwanted movements. So when people have Parkinson's, you see the shaking, you see the pill rolling, right? You see the actual tremors when they move. Well, those are all unwanted movements that will be inhibited in all of us without Parkinson's due to the activity of those neurons in the substantia nigra. As they release dopamine, they will inhibit those unwanted movements. Well, as those neurons release less and less dopamine, those unwanted movements will be more commonly seen. So why don't we just give the people dopamine, right? If you've ever seen the movie Awakening, that is what they did. They give people just a high dose of dopamine, right? Now, when you give people a high dose of dopamine, a little bit of dopamine can cross the blood-brain barrier. But you have to give them such high doses. Dopamine is in the same class of neurotransmitters and hormones as epinephrine and norepinephrine. In other words, they're beta adrenergics. They activate your sympathetic drive. So you give somebody that high dose of dopamine, it can sometimes affect their heart rate and their blood pressure. Not good. So what happens? Well, dopamine at high doses can cross a blood-brain barrier, but you have some massive side effect, right? In other words, dopamine is too large, right? And it had a hard time getting through the blood-brain barrier. In order for it to get through, you had such high numbers, high doses, that you have some very bad side effect. So nowadays, we don't give dopamine to people with, right, at the very first stages of Parkinson's. We give a precursor that can cross the blood-brain barrier. We give them L-DOPA. L-DOPA then can cross the blood-brain barrier, get picked up by those dopamine-releasing neurons. L-DOPA is a precursor. So as it gets picked up by the neurons change it to dopamine. And then the neurons can release dopamine and hopefully inhibit the unwanted movement. But that is just how specific the blood-brain barrier is. It will allow L-DOPA to cross, but not dopamine, right? So it's very important to limit the movement of molecules from your circulatory system to the brain and spinal cord. Now, the other cells that we see that's part of the neuroglial, epidermal cells. Epidermal cells, they line openings, empty pockets of our brain called ventricles. So we think of the brain as just one big solid mass. In reality, it isn't. There are openings, there are, right, ventricles that are filled with fluid that's inside the brain. The ventricles that are deep inside the brain will produce cerebrospinal fluid that allows us to bathe the brain, right, and to allow the brain to float above the skull. The skull, if you guys have seen a real skull, you'll know that the inner basin of the skull has a lot of very sharp bony edges. Now we have, later on we'll talk about, connective tissue that covers the brain, yet the bony aspect of the skull is so sharp, it can probably cut through that and damage the brain. So we don't want the brain sitting directly onto the meninges and onto the skull and all the bone that makes up the skull. Too sharp. Brain is too fragile. So what do? We have a fluid, cerebrospinal fluid, that allows you to bathe the brain in this nutrient-rich environment, high sodium, right? High potassium environment, high glucose environment, and it allows the brain to float so then it's not just sitting on that sharp skull basin. Now, where we produce a cerebrospinal fluid is in the openings in the brain, right, called the ventricles. The ventricles of the brain are lined with blood vessels, and the blood vessels contain these epidermal cells. These epidermal cells allow us to filter the blood vessels called the choroid plexus. So the choroid plexus are the name of the blood vessels. Blood goes through the choroid plexus and then gets filtered by these epidermal cells. As it gets filtered, a little bit of salt gets through, sodium, potassium, a little bit of, right, a little bit of glucose gets facilitated, diffused through, and water goes through, right? It then produces CSF, the cerebrospinal fluid, then goes through a series of connections between the different ventricles of the brain, eventually then draining out. As it drains out of the internal structure of the brain, now that CSF will drain out, and then it will then bathe the external component of the brain, allowing then the brain to float on the cerebrospinal fluid. So these epidermal cells, important because they will filter blood going through the choroid plexus to produce cerebrospinal fluid. They do this in areas that we call the ventricles. We're going to see that there's 2 lateral ventricles on each hemisphere of your brain, one on each hemisphere. Then they're connected to a central ventricle called a third ventricle, which is really deep in the brain itself as well. And then eventually to a fourth ventricle near the connection between your medulla oblongata and pons and with your cerebellum. It is in that fourth ventricle region where a little bit where the CSF starts to leak out, internally leak out from the inside of the brain outwards. So there are little openings that allows the CSF you form to leak outwards. Then it's going to bathe the brain and the spinal cord with that CSF. Then other types right here. Neuroglial cells that we just mentioned includes astrocytes, epidermal cells. The next one are microglial. Microglial cells are specialized macrophages. They respond to inflammation. They allow you to phagocytose necrotic tissue and whatever foreign microorganisms gets through the blood-brain barrier. So these are our immune cells that is there to fight off hopefully any infection that can get through the blood-brain barrier. The oligodendrocytes right here. Oligodendrocytes are going to be the name of myelin sheathing cells found in the central nervous system. Now, what we're going to see is this, right? What would happen if you are driving, and as you're driving, there's a slight delay from bringing the information from your eyeball, the light information from your eyeball into the optic nerve. What happens if there's just a little millisecond or even a couple millisecond delay? And now, as you're driving and going 60 miles per hour on a freeway, you're not receiving that information from your eye in real time. Well, that's bad, right? So what we need to do is to speed up action potentials as fast as possible. So then there is no delay. Because if there is a delay, well, you're going to be in serious trouble if you're going fast. So what we do in the body to speed up action potentials is undergo a process called myelination. Myelination is what we see here. We have multiple layers of fats, right, covering a central axon. What happens then? Well, if you have fatty layering covering the axon, sodium can't get past that fat. There's a physical fatty barrier in the way. How can sodium enter the axon and trigger an action potential then? It can't. So regions that are bare, little cracks and crevices that are bare, where you have one myelinated sheathing, right, and then another one over here, but there are cracks in between. Those regions are called nodes of Ranvier. Only in the bare regions will you have action potentials. What happens? Well, you bypass all this area. So then this area that are myelinated no longer have to propagate the action potential. It gets bypassed. So from here, one bare region, and then you jump all the way over to the next bare region. Then you have an action potential here. Then you jump all the way to the next bare region, bypassing all the layers that are fully myelinated. Allows you to speed up action potentials tremendously. Now we'll come back to the myelin sheathing in a second, but in the central nervous system, oligodendrocytes will myelinate. And as they myelinate more than one axon at more than one location. That's going to be very important, right? Oligodendrocytes will myelinate more than one axons at sometimes more than one location. Next, right? So before, those were the cells that we mentioned that are part of the central nervous system. Now we're going to talk about cells of the peripheral nervous system, including Schwann cells, right? So these are big ones: Schwann cells, satellite cells. Schwann cells, or neurolemocytes, wrap around one portion of one axon, forming a much more complete myelin sheathing. I want you to think of this as like jelly rolls right here. Jelly rolls covering the axon. What happens? Well, the thicker the jelly roll, the more complete the wrapping around the axon, the more likely you're going to bypass that region. So in the Schwann cell region, each Schwann cell only wraps around one part of one axon. So here's one Schwann cell here. Well, to have another region fully myelinated, we need another Schwann cell. Very different than what we saw in the central nervous system, right? Where you have one oligodendrocyte that can myelinate multiple axons. Here, one Schwann cell, right, only wraps around one part of one axon, and that's it. Now, what happens though is that the wrapping of each axon is more complete, and you're more likely to bypass those regions that are fully myelinated. And then the space between these two Schwann cells, this crack, that's a node of Ranvier. That's the only region where we'll have action potentials. During development, as cells grow around the axon, cytoplasm is squeezed out and multiple layers of cell membrane wrap around the axon, and then the cytoplasm, right, is seen at the most external region right here. Cell membrane primary phospholipids, the greater the wrapping, the faster the speed or conduction of an action potential. Satellite cells are also found as part of the peripheral nervous system. They surround neuron cell bodies in sensory ganglia and provide support, nutrients, and sometimes repair as well. So these are all the different types of neuroglial cells. Know the differences. Really good for multiple choice, really good for matching questions. Now, next thing we'll talk about is myelination versus cells that are and axons that are non-myelinated or unmyelinated. Now, myelination of axons occurs and starts after birth, right? When the baby is first born, the baby usually has not myelinated any of those axons yet, right? Now, that doesn't mean that the nerves haven't formed. They have formed. They form early on in gestation. It's just that the nerves are not functionally active yet. In order to activate them, you need to trigger action potentials in the nerves. And in some of these nerves, right, as action potentials will slow down because there's no myelination, the slowing down the action potential, right, will make it impossible to cause a real intended motor movement. So we believe that with infants that are newborns, we call neonates, their vision is about 6 to 7 inches. They can only see clearly about 6 to 7 inches. So why? Why can they only see that far? It's because of the optic nerve, even at their age of a couple, hours to a couple days old, right? What happens is that the optic nerve hasn't fully myelinated. Myelination requires lots of fats. And that fat can be now coming in, right, from breast milk or from formula. By the way, that's one of the reasons why, if you look at formula, they always tout the kind of fats that they have. And they talk about how it allows for brain development. What it really allows for is myelination. The fats, the AHA, the FSHA kind of fats, right? Those are all readily incorporated in the myelin sheathing. So myelination is requires that fat. Now, breast milk is the best because the fats that are found in breast milk not only will be incorporated into the myelin sheathing, it's also more easily absorbed. And within the fat, you also have immunoglobulins as part of breast milk. So then you have that additional aspect of, right, immune protection because you have antibodies in mom's breast milk. Now, myelinated axons, myelination starts after birth from the top-down sequence. That's a big one, right? There's a sequence of development that pediatricians want to see. For the most part, pediatricians understand that every parent's going to be proud of those milestones, right? Oh, my child is able to walk at 10 months. Oh my God, you're so talented, right? So usually pediatricians really don't, I wouldn't say they don't care, but they're not worried if you don't walk within 16 months, within a year, right? As long as we hit the milestones from that top-down fashion, then we know development is occurring. And then everybody's going to develop at their own pace. So with myelination, it starts off from the top down. So baby is going to myelinate the optic nerve and the brain, right? And the peripheral nervous system in the brain, next to the brain, the cranial nerves. So you're going to myelinate that. As you myelinate the optic nerve, you start being able to see a little bit further. Then you're going to myelinate the spinal nerves that will then go to your erector spinae muscles in your neck, semispinalis capitis, right? The splenius capitis. What happens? Now baby is able to hold their head up as they attain that function because they myelinated the spinal nerve segments from the top down, right? As you myelinate the spinal nerve sections from the top down, next segment is going to be the brachial plexus. So what happens? When you have a 1 or 2 month old baby, if you put your finger in their hand, they immediately squeeze as a reflex. It's not an intended movement, right? And usually, parents, they kind of make you feel better by saying, oh, he likes you. That's why he's squeezing his finger. It's a reflex, right? Up until about 3-4 months, it's a reflex. If it's within the first couple of weeks, they can't even see you, much less like you. So they're not going to be squeezing because they like you, right? But it's okay, right? They probably love you. It's okay. So what happens is after you start to myelinate from the top down, you start myelinating the nerves that form the brachial plexus. And now you start having more intended motor movements of your shoulder and then your upper arm and then intended movements of your forearm and then your hand and your fingers, right? Eventually, you have a pincher grasp that develops. Again, top down, right? And then what happens? If you have a 3-4 month old baby, they can hold their hand, all right? They can move their hand and arm with intended movement. They can have their heads, up straight. They can hold their head up now. If you put that 3-4 month old baby and you sit them up on the couch, right? They usually fall over. Why? It's because they haven't fully myelinated the rest of the erector spinae muscles. Again, top down. As the brachial plexus get fully myelinated, then we'll myelinate the spinal nerves that will hold and activate the rest of the erector spinae. So then they can sit up for a prolonged period of time. And then what happens? You myelinate the lumbar plexus. Now they can move their lower limbs with intended movement and strength. Now they can crawl, right? They can roll over and then crawl. And then they can stand up for a longer period of time. And eventually they can walk, right? Again, continuous top-down myelination allows us to have continuous top-down movement control. Now, with myelination, we have this, right? In terms of myelinated axons, the myelinated axons, the myelin sheath is not one continuous sheath. It's made out of multiple schwann cells that will have tiny breaks, right? Those breaks in the middle where there's no myelination are called the nodes of Ranvier. These are gaps between the myelinated section. It's the bare regions. And this is the bare regions where now sodium and potassium can enter and exit. Sodium enters to depolarize, Potassium exits to repolarize. This is regions where the action potentials occur. You should complete myelin sheath development at 1 year age. That does not mean that all babies should be walking at 1 year of age, right? It just means that you've had the actual anatomy, right, there, and now you're fully myelinated. The coordination of when to take the step so then you don't fall, that takes time, right? So as long as an infant or the baby, at that point, now not an infant's a baby, is able to walk within 16, 17 months, you're in pretty good shape, right? Even 18 months. If it's about 20 months, then doctors start being a little bit worried because of that delay. But usually, right, even then, eventually, almost all babies learn to walk. Degeneration of myelin sheathing can occur in a couple of diseases. The most common one, all right, is still uncommon, but it's the most common one of the myelin sheathing disease, is MS. MS, multiple sclerosis, do not get that mixed up with MD, muscular dystrophy, right? Muscular dystrophy, especially the Duchenne's form, is almost always fatal and almost always affects males. This is different. MS is an autoimmune disease where we have autoimmune antibodies that starts attacking our own myelin sheathing. How can you tell? So then you don't hopefully get these two diseases confused. It's simple. MS for multiple sclerosis, MS for myelin sheath. Right? Now again, what's the point of this myelination? Is to speed up action potentials. The faster the action potential moves, the more likely you're going to keep stimulating the muscle so then you can have coordinated movements. Right? Now that also happens in sensory. The faster we bring the information into the brain, the faster we can process it, right? The better it is. Now, the big thing about myelination is this, right? And this is, you're wondering probably is why don't we just have everything myelinated then? If all myelination allows us to do is speed everything up, how come we don't myelinate every part of the central nervous system? In the brain, it should not just be, it should just all be white matter, right? Well, the thing is, if you're moving, right? Let's just say an example, right? If I'm running a mile, it makes sense I want to run faster so that I can finish that mile, right? So speed becomes important then because it's such a far distance, right? How about if I'm only taking a step from my couch to the TV to turn it on because I've lost the remote? Well, usually if I'm only taking a couple steps, does it matter how fast I'm moving? No, right? I'm only taking a couple steps. Then I'm going to sit back down. So what happens? Depending on how far the action potential has to move, you might need to speed it up. Sciatic nerve, very long. You might need to speed up that action potential. So then you can bring that information in fast, right? Or bring that information out so that you can contract the muscles because it's so long. But if all we're moving is carrying that action potential, just a tenth of a millimeter from one interneuron to another, does it matter how fast that action potential moves? Not really. It's such a short distance that in terms of myelinating, it would add more energy to maintain it than to just have it fully unmyelinated just because it's such a short distance. So the shorter this distance, the less myelination plays a role in its transmission. Right.